Good morning, all. Hope you all are doing good. So today we are going to discuss about the human cell. So as you can see, this is the human cell, and there are a lot of components inside the human cell. Each component performing a different function. So these components are called as organelles. So organelles are like little organs which are present inside the human cell performing distinct functions. So it is no similar to our human body where we have different organs like our heart, lungs, stomach, all performing different functions. Similarly, inside the human cell, we have different organelles, that is how they are called, performing different functions. So, a human cell is the smallest unit of the human body. So, the whole human body is built upon this smallest unit cell. So, when the cells multiply and they grow, they become human beings. So, we will see how these cells were formed in the first place and why it is important to even study about this human cell at this small microscopic level. We know the organs how they function, so what is the use of even trying to study the cell which is so small and trying to understand how the components which are further smaller than the cell itself are performing inside the human cell. So, the reason why it is important is if you understand the cell at the microscopic level, you will be able to understand the organs at the macroscopic level. So, if you understand why this cell is behaving like this at this small level, you'll be able to understand why we get diseases, how these diseases can be cured, how they can be prevented, because everything happens at the cell level. The cells combine together and work together as an organization, and we get the output, which is our heart or lungs or stomach. So they combine together and perform the various functions inside the human body. So, how this small unit cell was found in the first place? So, the cell was formed from an atom. So, if you see, atom is the smallest unit of matter, smallest unit of non-living thing. So from atom, many atoms combine together and they form molecules. So molecule is nothing but when one or more, two or more atoms combine together to form molecules. From these molecules, what happened was they combined together and formed bigger molecules. Complex molecules were formed, different molecules combined together, and they formed something called macromolecule. So examples of macromolecule can be your sugar, proteins, which are present inside your body. So these macromolecules, they combine together and they form with something called organelle, which we were saying these are the parts which are mini tiny organs which are present inside the cell. So these organelles were formed, and these organelles combined together they formed the cell. So that is how this cell was formed. Now, after this cell, if we go further, how we human beings were created. So, from cells, the cells with the same function, they combine together to form tissues. So, different cells form different tissues, group of tissue. Cells is called tissues, same functioning cells. Now, different types of tissues combine together to form an organ. It can be your heart, your stomach, your lungs. So, here we are seeing the stomach. Now, these different organs combined together, they form the system. Now, here we are seeing an organ system. Example, here we are seeing digestive system, like your stomach, your large intestine, your small intestine, your esophagus. So, all combined together, they form the digestive system. Now, similarly, all the systems were formed and an organism was formed, that is, human beings. We were formed with all different organ systems like our respiratory system, digestive system, nervous system, our heart, circulatory system. So, all these systems combined together and an organism was formed. So now we are going to discuss about this cell in detail. So you can see how small is the cell. This is the smallest part of our human body, and we are going to study about this cell now. So this cell is what is in a bigger view how this cell looks. So if you see, the cell contains a membrane outside it, which is the cell membrane, which is semi-permeable. Means it will allow some things which are useful inside the cell and which are not useful or bad, it will not allow it to enter the cell. Similarly, it will send out which is not good for the cell, and it will take in what is good for the cell. So, this cell actually looks like one squeezy ball with water filled inside. So if you see, it will be like this one ball with water inside it. So that's how this cell looks. So when we cut open that cell, we see all these organelles inside it. So the first one you can see is the nucleus, the bigger one. So nucleus is, you can see, it's spherical in shape, and once we cut open the nucleus, you can see the nucleolus, the inside the dense part, this nucleolus. And we also have this something called this chromatin inside the nucleus. Okay, so now this nucleus is the brain of the human cell. Brain means it controls whatever has to be done inside the cell. Like our brain is there, our brain tells us to walk, we walk, talk, we talk. So that's how here for this cell, this nucleus acts as the brain of this cell. So this nucleus contains that brain we are telling, right? That brain is nothing but your chromatin. Chromatin is nothing but your folded DNA. So DNA, you would have heard about DNA. From DNA we get the same features, heredity, how the genes get forwarded from one generation to another. So this chromatin is nothing but your folded DNA, which is no, folded and kept inside this nucleus. So that is your chromatin. Next is your nucleolus. Nucleolus is the dense structure which is this does not have any membrane as such. It is just dense part which is present in the nucleus, and what it does, it creates the ribosomes. So the ribosomes, these circular structures which you are seeing, small ribosomes. So ribosomes are produced here from nucleolus. So that is the work of this nucleolus. So nucleus work is giving instructions when to do what. Nucleolus is manufacturing of the ribosomes. So what are these ribosomes? Okay, you see these ribosomes is pink small ball structures which are here. So these ribosomes are the what to say, they are like the cook which prepares proteins, which can be used for performing various functions like construction of organs or repairing of organs, or for any other purpose. These proteins are manufactured by ribosomes. So ribosomes are very useful for manufacturing proteins which are used in the construction of the various other organelles inside this human cell. So that is the use of the ribosomes. Next, the other part which is present is attached to this nucleus. If you see, these are endoplasmic reticulum. So you can see here, this is the endoplasmic reticulum, which are divided into two: rough endoplasmic reticulum, okay, and these tube-like structures, these are smooth endoplasmic reticulum, okay. So if you see here, the rough endoplasmic reticulum, you have the ribosomes attached to that. So these are nothing but the flat network pathways. You can see it's like that maze. The flattened walls are there, and in between that maze, we have a pathway. So that is endoplasmic reticulum. And since the ribosomes are attached to this endoplasmic reticulum, this is called rough endoplasmic reticulum because it gives a rough look because of that. So now what is the purpose of this rough endoplasmic reticulum? What is the purpose? Whatever ribosomes are preparing, no, the proteins, they fold that proteins, whichever are attached here, they fold that proteins into useful forms which can be sent to other organelles to repair their part or use in whatever form it requires. So what this endoplasmic reticulum, the rough one, does is it folds the proteins and it packs it into some vacuum bubbles called vesicles. So it is called vesicles. We have vacuum bubbles. They are surrounded with membrane and they are released into this uh, space inside the human cell. So this entire space is filled with what do you call cytosol, which is this is called nothing but cytoplasm, where you'll have semi-fluid liquid. So semi-fluid liquid, so cytoplasm, semi-fluid liquid is filled. I told no, it is filled with some water. So that is semi-fluid liquid, which is called cytoplasm. cytosol, that liquid is called the entire space along with its organelles is called cytoplasm. So inside this cytoplasm, it sends these proteins. So the use of this rough endoplasmic reticulum is it folds the proteins which are manufactured by these ribosomes, and it packs it into some membrane. It covers it with membrane and leaves it in cytoplasm so that it can be used by organs going forward. So what happens is from here, whatever vesicle is sent. So if you see endoplasm has sent the vesicle, this enters the next organelle, which is nothing but your Golgi apparatus. So Golgi apparatus, what it is doing is it takes the vesicle or the protein covered with membrane which is released by the rough endoplasmic reticulum, takes it and it adds whatever is necessary or modifies the protein, and it releases the useful form which can be used by organs for their repair or any other purpose. So it acts like that post office. So you are sending letters, it's no sorts it, combines it, modifies it, and gives output, and it supplies that letters to whichever organelle it has to be sent to. So Golgi apparatus does that post office work. So next organelle is your same endoplasmic reticulum, but this time it's smooth endoplasmic reticulum. If you see the smooth endoplasmic reticulum, here it does not have any ribosomes attached to it. See, it is smooth. So that's why it's called smooth endoplasmic reticulum. So this does not produce any proteins. It produces lipids, the fat content called lipids. So so if you can see this smooth endoplasmic reticulum, as I told, it produces lipids, and that lipids are again packed into membranes, and they form vesicles, and that can be used by the organelles for further purpose. So next organelle which you are going to see is the centrosome. So if you see the centrosome, these are the centrosomes which you are having two centrioles which are cylindrical structures which are perpendicular to each other. So these are uh, useful, or they will be useful when the cell goes or undergoes division. So when new cells need to be formed, it undergoes mitosis, which is division of cells into two cells. So that time this centrosome will be useful. So that is the function of this centrosome. And next, if you see, next comes this mitochondria. So if you see here, this is mitochondria. So mitochondria is called the powerhouse of the cell. That is, it is the power plant. So it generates the energy which can be used by rest of the organelles for doing their function. So mitochondria has its own set of DNA with which it can divide itself and multiply. So Mitochondria is called the powerhouse of the cell. It uses the nutrients which are absorbed into the cell and oxygen to produce some small batteries called ATP. So it basically generates power. So that energy which is generated by mitochondria is used by all the organelles. So that's why it is called the powerhouse of the cell. So if mitochondria does not produce any energy, nobody will have an energy to do anything in the cell and the entire cell will die. So mitochondria is that much important for the human cell. So next comes your lysosome. So lysosome is like your scavenger which removes the waste from the human cell. So sometimes some organelles inside the cell die, or there are some bacteria which are present inside the cell. So that time this lysosome comes into play. So this lysosome, what it does is it has some digestive enzymes which what they do is digest any unwanted materials. So this has protective layer around it. If it does not have protective layer around it, the digestive enzyme, which is very acidic, will kill the entire cell itself. So that's why it is sometimes called the suicidal bags, because if its contents are released outside into this cell, the entire cell will die. So that's why it has special protection to prevent the digestive enzyme from leaving this lysosome. So whenever it sees any organ is uh, failing and it has to be killed, or any debris is there, it goes and combines with that organelle inside a vacuole, or it takes the waste inside a vacuole. So vacuole is nothing but one bubble like formation inside this cytoplasm where the if even if it releases contain digestive enzyme, it remains inside this vacuole and it does not escape it into the cell, because as I told, if it escapes into the cell, it will kill the entire cell. So that's why this combines with whatever debris is there which has to be recycled or cleared. It goes inside this vacuole and it combines with that waste or debris and it digests that materials by releasing its digestive enzymes. So that is how lysosomes work. So they are basically the scavengers. Next is the peroxisome. So peroxisome is an another form which also does the work similar to lysosome, but it is called no specialized lysosome, we can say. It does work which lysosome cannot complete, the peroxisome can complete. For example, when hydrogen peroxide is present inside the cell, it helps to remove that toxins from the human cells. So we can say the peroxisome is like a specialized force which the lysosome cannot do. It will do that specialized scavenging work by peroxisome. So this is about peroxisome. Next is this filaments, which you can see the blue color filaments, microtubules, microfilaments, which are present. So these are hollow structures, thread-like structures. So these are present throughout your human cell. So this give the structure to the human cell. Like no, we have all iron rods laid inside the building to give it no strength. So that's how these filaments are present throughout the human cell, giving it a structure to keep the organelles in place from moving here and then, or for the organs to move from one place to organelles to move from one place to another. They use this microfilament microtubules which are present inside the human cell. So that is all about the human cell. Hope you would have understood so far. So now the reason why we are studying about this human cell, it will be very useful to understand at the microscopic level. So, for example, if you see here, in case the mitochondria does not work properly in the cell, what I told, it will the cells will not have the necessary energy to work, and it will lead to the cells death ultimately. So if this one cell dies, the same chain reaction, if it continues, what happens is your organs lining or your organ itself may start failing. So that is why just one human cell is so important. The cells can repair some things on their own, but sometimes when they are unable to repair, they go into this suicide, where this lysosome comes into play and the cell kills the entire cell itself. So, if any of the organelles inside the cell malfunctions, then you can see, for example, like you have cancer. That happens because the DNA inside the cell or the brain of the cell, the nucleus, it loses control over the cell, and the cells divide unlimitedly without stopping. So that's how this human cell influences the organ functioning. So hope you have enjoyed today's session. If you like this video, please like, share and subscribe. Thank you all.